Thank you for avoiding the tempting siren song of the pool and coming back to this ballroom for another session. I appreciate it. I thought by way of broader introduction, I'd tell you a little bit about the interests of our lab and how we got into this field. The big picture question I'm interested in is the genetic causes of congenital anomalies of the brain and face. And we use a combination of mouse genetics and human genetics to identify these unknown genetic underpinnings. And then we leverage the power of mouse models and/or cellular models to understand the molecular mechanisms. And then if we understand the mechanisms, can we use those tools to think about clinical interventions? So as you might imagine, a broad approach like that takes us in many different directions, and these are some of the areas that we think about, including congenital disorders of glycosylation. And then what I'm going to tell you about today is some of our efforts on AAV9-mediated gene replacement of PIGA. So that's the big picture that guides us day to day. How did we specifically enter this field? So in our mouse mutagenesis efforts, where very briefly we chemically mutagenize mice to accelerate the process that happens naturally by many, many fold. And we just ask, what are the new genes that come out of this forward genetic screen? And in the early days of the screen, one of the alleles that I got was this allele, which has a number of different phenotypes, including cleft lip, cleft palate, and exencephaly. And when we cloned the causal gene, we found out that this was a variant in PIGA-2, which is in the GPI anchor biosynthesis pathway. At this point, I was among the unwashed that Professor Fries talked about on the first day and had to learn this pathway, or at least get familiar enough to talk about it. So GPI anchor biosynthesis, you've seen this a couple times, is a multi-enzyme reaction that adds a GPI anchor to about 150 different proteins in the cell to facilitate attachment on the cell membrane. So our route into this was at PIGA-2, which is here late in the pathway, involved in remodeling of the anchor. And at the time, I had a very talented MD PhD student in the lab who recognized that there was a lot we didn't know about the embryology of this whole pathway. And he suggested that rather than focus our efforts on PIGA-2, given how much we didn't know as a field, let's just go think about the entire pathway. So he persuaded me to get comfortable and think about PIGA, or PIGA, which is one of the first steps in this pathway, this complex that starts the GPI anchor biosynthesis. So I'll thank the previous speakers for two things. Tracy told you all about Cre-Lox genetics and the X chromosome, so remember everything you learned just before the break, and then Professor Lai talked about gene therapy. But the reagent we used to study PIGA was indeed a conditional allele of PIGA. So a mouse model completely lacking PIGA in the male situation is embryonic lethal very early in development and not amenable to studies that we would be interested in pursuing. There was an existing flox allele. So again, when you are able to drive Cre recombinase, you can specifically ablate a targeted or floxed gene in any way that you desire. So again, following almost exactly the same strategy that Tracy described before the break, we've used Cre-Lox to ablate PIGA. And of course, that results in two classes of mutants, if you will, the males who have no PIGA, and in this case, I'm only going to talk about brain-specific ablations. So the males, or what you'll sometimes see referred to as hemizygotes, have no PIGA anywhere in the brain. And the females are a mosaic, again, depending on if that random X inactivation process inactivated the healthy allele or the chromosome that did not have PIGA by virtue of the LOXP ablation. So these early studies, we first went back and asked, what is the consequence of removing PIGA throughout the nervous system using a nestin Cre? And this picture down on the bottom is a mouse reporter of nestin Cre activity. So this is a mouse around embryonic day 12. The blue represents where the Cre is active, and it's essentially throughout the entire nervous system. So that was the first experiment that we did along these lines, and we showed with Western blotting that this does indeed result in a major loss of PIGA protein in male and female mice. These mice had a number of interesting phenotypes, some of them very reminiscent of the patient population. They did not have striking microcephaly, so if you think that these knockout brains look a lot like control, you're absolutely right. You've passed Neurology 101. Those do not look very abnormal. The cerebella are somewhat smaller. The biggest finding for us is that male mice lacking PIGA throughout the nervous system, this is a survival curve like you've seen a few times where time is on the x-axis, percent survival is on the y. You'll see that a good chunk of the males, whoops, I did it again, sorry. About half of the males die on day 1 of life if they've lost PIGA throughout the nervous system, and we don't have any after 9 days. The mosaic females have a slightly different progression where they're not surviving to weaning stages either. These mice have a number of behavioral phenotypes as you might expect, and similar to what you've seen before, we see things like this hind limb clasping phenotype, this righting reflex, and then we're going to come back to this. So we've done the rotorod, which is the ability to remain on this rod that is spinning at increasing rates over time, and the ability to stay on the rod is a proxy for coordination. So these mice had a number of CNS phenotypes. One thing we noticed when we looked at some of the molecular analysis is that these mice lose or do not have a large portion of the myelin that is necessary for proper CNS signal transduction, as shown here by this staining in the male brain compared to the control. And another phenotype we saw was a disorganization of the Purkinje cells that you heard a lot about in the zebrafish talk closing off yesterday's session. We put a pin in that and thought a little bit about what we wanted to do next. And the nesting tree is a pretty broad tool. So we wanted to ask within the CNS writ large, which portions of the brain maybe are most important to have PIGA expression in. So we tried to dissect this out a little bit using 3 different Cree transgenics. So I'll summarize in one slide what happens when you ablate PIGA throughout the forebrain using the EMX1 Cree tool that you just heard about in the previous talk. We also asked what happens if you ablate PIGA in the white matter cells, again using that OLIG2 reagent you've heard about. And then we asked what happens if you ablate PIGA in the cerebellum using an engrailed 1 Cree. And very simply, if you delete PIGA from the forebrain and just the forebrain, there is a survival deficit in that the males now are not surviving. We lose about half of them in about 28, 29 days of life, which is a lot less severe than the nesting Cree-mediated deletion, and their weight is significantly less than litter mates. But the brain is structurally normal as far as we can tell. They do have the loss of myelin like we saw with the nesting Cree. And if we look at some of the architecture, as previous talks have shown, we can show fairly subtle deficits in CNS architecture from just this forebrain deletion. Next, with the OLIG2 deletion, we see a mild decrease in recovery of the animals without PIGA in the white matter at weaning. And as expected, we see these myelination defects. But again, this is much less severe than the nesting Cree ablation. However, with the cerebellar ablation, the male mice are basically never recovered after day 7. And the cerebella of these mutant mice are significantly smaller than controls here. And we think part of this has to do with the failure of these Purkinje cells to properly migrate and form the supporting structures for that granule cell proliferation, which ultimately contributes most of the mass of the cerebellum. So the answer to that question, if you had to put PIGA anywhere, where is it? The answer is, if you're measuring simply survival, the answer is the cerebellum. And we thought that was actually pretty interesting. We were doing that because in parallel, we were thinking about gene replacement therapy paradigms. And in the event that a broad delivery didn't work, we thought this was going to be important to understand. I'll switch now to our results with that gene replacement therapy. So I moved to Nationwide Children's about 5 years ago, and part of the rationale was to get to know our colleagues in the Center for Gene Therapy better. So multiple FDA-approved therapies, some of which you heard about earlier today, came from our institution, and that was a space we wanted to get comfortable with. I had no idea how proximate I was going to be because Catherine Meyer was at that time at our institution. She's since left to start her own company. She was engaged in a collaboration with Taro Kinoshita to do exactly this, to test the ideas of gene replacement therapy for PIGA. Given that we were in the same building and with the blessing of Professor Kinoshita, we have carried on this work. So very similar to the workflow that Tracy showed you, we are injecting AAV9, in this case AAV9 human PIGA, into the mouse cerebral ventricle at day 1. And we're doing some of the same measures, looking at increases in survival and changes in behavior and performing some molecular analyses of those animals. And I'll just hit some of the high points of that here. First and foremost, we see a major increase in survival in the female mice. Again, untreated mice We lose most of them by day 28. We've tried 2 doses of this AAV9 human PIGA, and both show a significant increase in survival time, and we are seeing a dose-dependent effect on the length of that survival. We never thought we would go this far, but we decided based on the strength of that data just to ask what would happen with the male mice. Again, these are now missing PIGA throughout the entire nervous system. Untreated animals with that condition, 50% lethal by day 1, never make it to day 10. We're seeing a remarkable increase in longevity of even these male mice with the human PIGA replacement therapy. These animals were sacrificed for molecular analysis, but we took a different cohort to ask how long would this survival last, and we have preliminary evidence that this will be a sustained effect. So again, another set of controls, untreated females, lethal by about weaning. Again, a dose-dependent response. So for instance, at the higher dose, we're seeing about 3/4 of these females are surviving at 6 months. And to me, again, even more surprising, we see a robust effect with the male mice where we have about 1/3 of them surviving at 6 months. So I submit that survival is a pretty good biomarker. So I think this is encouraging, and this is something we want to pursue further. So how healthy are these mice? So looking at that 6-month time point, just simply weighing the animals, we can see that with the females, the treated females don't weigh quite as much as the controls, so they're a little bit smaller, but they're generally healthy. And again, to me, continuing to surprise me, the males are indistinguishable in terms of body weight at 6 months of age from the control animals. Some of the molecular analysis that we've pursued is to look again at those myelin tracts in the animals. So I'm showing you here a higher magnification of the cerebellum. These are the Purkinje cells. They express PIGA at relatively high levels, and in these mosaic animals, there's very much reduced expression of PIGA in the Purkinje cells. And I'm showing you the sustained effect here at 4 weeks of the PIGA expression in those Purkinje cells in both the females and the male mice. Using fluoromyelin instead of the stain we used before to look at the health of the myelin tracts, again, you can see a significant rescue of myelin in the treated animals compared to the mosaic. There is no male to show you because the untreated male does not survive to this point in time. What about longer term? So again, these exact same markers in the same layout, PIGA expression in the Purkinje cells at 6 months of age, and in the males seems to be sustained. The myelin tracts may not be completely normal. This is the cerebellum again. Those are significantly rescued levels of myelin. And if we just ask quantitatively how much PIGA is there in I'm trying to remember exactly what this lysate is from. I think this is from cerebellum. Even at 6 months after a one-time dose at day 0, I challenge you to tell me which is the mutant and which is the wild type. So we have a sustained increase in PIGA expression. What about behavior? So this is the rotor rod that you've seen before and I described earlier. In terms of the females, so you want to see the treated females are in blue and the treated males are in this purple color. The females are statistically indistinguishable from wild type. I will not say that they're indistinguishable, but they are significantly improved. And then the males, again, untreated males don't exist. These are performing virtually identically to the wild-type males, even at 6 months of age. As just another marker of CNS development, this hind limb clasping score also shows a significant rescue, here shown at P21. The last piece of molecular data I'll share with you is this attempt to understand what happens to the GPI-anchored proteins themselves. So we're not going to talk about glycosylation assays. We're in a different part of CDG biology. We've been able to take advantage of this fact that one can use this alpha toxin, which binds GPI anchors directly, and biochemically pull down the alpha toxin and ask what GPI proteins are present. And then one can perform the mass spec to figure out that protein population. So we've done this, and we see a number of proteins through this AT capture or alpha toxin capture that are lost in either the males or the females. and there's 12 that are lost in both. And we wanted to ask, how do those specifically respond to the AAV9 treatment? And what we're plotting on this graph is the untreated females, the wild-type females, the treated females in this green color, untreated males in this orangish color, and treated males in purple. And we're just looking at levels of these individual GPI-anchored proteins relative to wild-type. So as you would expect, the untreated animals, the yellow and the orange, have significant loss of these proteins. And in almost every single case, specifically looking at GPI-anchored proteins, we see a major rescue of the levels of these proteins in the treated animals in the green and pink bars, respectively. So it's not a biomarker per se, but we're directly looking at how much rescue we're achieving to the ascribed function of PIGA. So those are the pieces of data I wanted to share with you today. What we're thinking about doing going forward and how I want to learn from the people in this room is summarized on this slide. So we also want to think about what are the time windows for treatment. We're treating at day 0, which is basically late second trimester, equivalent of the late second trimester in human pregnancy. We want to ask, how much rescue can we get if we treat later? That's not a viable path for males in this case, but if we treat those females at, say, day 10, how much rescue do we get? Conversely, thinking about a world in the future where prenatal diagnosis is, I think, going to be a reality, if we can get in and deliver this gene therapy back in the mouse, say embryonic day 14, sort of early stages of organogenesis, how efficient or how complete can that rescue be? And that's something that is experimentally tractable in mouse, and I think in the future will be tractable in humans as well. We've only tried 2 doses. We should probably do a larger dose response curve. We have a meeting in 10 days to go over the results of a single-cell RNA-seq experiment that we've performed with multiple goals in mind. One is to think about really more deeply interrogate the molecular phenotype in these mosaic females versus controls. But we also want to leverage this technique to look at the biodistribution of our viral product. We've done other injections with just GFP, and in our regimen we see broad expression of GFP over time. Our redelivery of human PIGA is not tagged in any way, so we can't look at that specifically. But this is human PIGA in the mouse PIGA brain. So what we've done with this single-cell experiment is spike in human probes that 10x tells us will be specific to the human transcript versus mouse. So we should be able to ask, within the context of this experiment, what is the biodistribution of our product? So I think these are really promising studies, but in some ways these are not ideal for the patient population. We are deleting this gene in tissue-specific ways. That's a little bit different than having an allele that is not a full deletion, but is a hypomorph or a reduced functional allele everywhere in the body. That is something we've tried to do with 2 different cores at different universities and in our own lab. It turns out that females in the mouse, females carrying pathogenic variants do not survive. We think this is because in the human population there is skewed X inactivation. So female carriers are, as far as we know, completely healthy, and we think that's because through a mechanism I certainly don't understand, and I don't know if it's understood at all, is that the body tends to inactivate the pathogenic allele. That does not appear to be the case in mouse, so we would attempt to make a germline female carrier. That is not possible in the mouse, so we have to think about a more sophisticated approach, some sort of a conditional knock-in of the poison exon, which we will continue to pursue. Using both those mouse avatars as well as continued Cre-Lox approaches, we want to think about learning about the role of PIGA in other tissues. Of course, this requires resources. There are some programs through the NIH to pursue this kind of study. These are the IGNITE and the URGENT proposals. We have submitted an IGNITE proposal that was apparently off by just a couple points, so we've resubmitted that and look forward to a positive review in July. And really what I want to think about together with you all is this a viable path forward? I think the 4 people in the world that are most qualified to have that conversation with me are at least in attendance today, and I can't tell through the blazing spotlight if they're here or poolside. But if you're some of those people, I really want to talk to you about, is this something we should push? What do we know from the patient population in terms of what our outcome measures should be? So I truly want to engage in that conversation. I'll be the guy who's about this tall, who's willing to buy you a coffee or an alcoholic beverage of your choice to discuss that, and I look forward to those conversations. Thank you very much for your time today and the opportunity to be here. Oh, the most important slide, and I blew it. Sorry, Andrew, 20 more seconds. I'd like to thank the people who did the work, especially virtually all of this was Jennifer Watts, a talented postdoc in the lab. Some of this funding came from this organization, and especially in these times, thanks to the NIH for current or recently completed funding to pursue the general goals of the lab. And now I'm happy to take questions. Thank you.